Hi everybody, my name is Precious Dialek and this is my presentation on ion channels and neuronal communications. So we have a couple learning goals that we'd like to achieve by the end of this lecture. So let's take a look at them so it will guide us as we take a look at our information. So the first thing that you should be able to do at the end of the lecture is to be able to explain how transmembrane proteins embed themselves onto the plasma membrane. You also need to be able to differentiate the different types of ion channels. You also need to be able to label the parts of a neuron and explain the concept of an action potential. You should also be able to explain the involvement of ion channels during neuronal communication. And lastly, explain how signal transduction works with the ion channel to elicit a response. So before we get into the main topic, let's just take a look at and review the properties of the phospholipid bilayer because it does dictate how the protein membranes behave in it. So the first thing that we need to review is the fact that the phospholipid bilayer is semi-permeable. Semi-permeable means that it only allows certain things through. So it is our cell's first layer of defense. So the cell needs to be very particular about what it lets in and what it lets out of the cell to keep our homeostasis, right? So things such as large molecules and charged molecules cannot easily enter the membrane. For example, glucose is considered a large molecule, and we do need glucose for cellular respiration. So it goes in through a special way. An example of a charged particle would be something like ions, such as calcium, potassium, chlorine, stuff like that, right? So those are not able to enter easily, whereas things such as gases, oxygen, carbon dioxide, and small amounts of water molecules can seep through the membrane easily. If we do need a larger amount of water molecules in the cell, there is a particular way also that they will enter. The second thing that we need to remember about the phospholipid bilayer is that it is empathic, which means that it has a polar portion and a non-polar portion. So if you guys take a look at the diagram there on the bottom, the pink circles that we see right here are our polar heads, right, which means that they like to interact with water, and our non-polar tails are these little green squiggles right here, which means that they do not like to interact with water. So the way that the bilayer is formed, the polar heads are the ones facing outside on both the intracellular and extracellular parts of the cell, so that the tails are nice and protected and don't have to interact with water at all. And then, of course, our phospholipid bilayer is called the fluid mosaic model because of all the different entities that are found in the membrane itself. So if you take a look at it, it is kind of like a hodgepodge of different items. We have cholesterol, which is signified by that yellow kind of hexagonal molecule. We have glycoproteins, we have transport proteins, which again is going to be the focus of our talk today, and a little bit of everything, right? So it is a fluid membrane, which means that those phospholipid bilayers and those proteins and cholesterol can move around. They're not cemented in one area. So now that we have had an overview regarding the membrane's properties, now we can learn about the proper way that the proteins must turn themselves in order to embed themselves in the membrane. So I'm going to move myself down a little bit so we can see the rest of the words. So when a protein gets embedded into the membrane, they actually travel through a different system, and this is called the endomembrane system or the secretory membrane pathway, right? So that means that when these proteins are translated, they are translated in the ribosomes that are attached to the endoplasmic reticulum, and then from there they go to the Golgi apparatus where they get modified. and then they get a special signaling sequence that allows them to know where they're supposed to go. In this case, they would know to attach themselves to the plasma membrane. We have two types of transmembrane proteins. We have integral and peripheral proteins. So let's talk about how each of them make their way into the membranes. So the integral protein, just like the word means, you integrate the protein into the membrane, so they are embedded in the membrane itself. So we have the alpha helix, the helical bundle, and the beta barrels. So what these proteins need to do is that they actually need to turn their R groups, their side groups, in a way where their nonpolar or their hydrophobic chains are interacting with the hydrophobic tails of the lipids. Right now, there is going to be a portion like up here at the tippy top where they're interacting with the heads. So they also do need to turn that part of their sequence so that it can interact with the head. So that's the same for any integral protein. In the peripheral proteins, that really just depends on how much is sticking out of the membrane. So peripheral proteins are usually located on the top or on the bottom of the membrane. They are embedded in the membrane, but just enough so that they don't float away, right? So if you take a look at this one has a lot more of its protein on there, depending on how deep it is in the membrane. Again, it needs to turn itself so that it can interact with the polar and or non-polar parts of the membrane, whereas this one right here is just kind of a couple strands that hold it down on the membrane. So these ones again would also have to turn their side groups the appropriate way. So now let's take a look at the types of transport proteins. We have two main types of transport proteins. The first one is a channel protein. So a channel protein is the one that we see that is usually open, right? And it uses passive transport. Now, when I say open, that doesn't mean that any molecule can just go through the membrane. Since they are proteins, they are still very particular about what types of molecules are allowed in and out through that specific channel protein. I like to compare the channel protein to kind of like a tunnel, right? A tunnel doesn't usually have anything like doors or gates blocking it, it's just open, but you still need to be the appropriate molecule to enter through that. So, for example, water, right? Water, like I said earlier, in very minute amounts can cross through the membrane. However, if we need a large portion of water, they need to go through the aquaporin channel. So the aquaporin channel allows large influx of water molecules in, and only water molecules, right? It cannot let in anything else except water itself. The second kind of transport protein that we have is a carrier protein. So a carrier protein is a little bit different because it's not always open. It needs to have a ligand bind through it, which will allow it to open and close. It does still follow the rules of diffusion, which means that we're going from high to low concentration. However, since the carrier is helping with the diffusion, this is then considered to be facilitated diffusion. So those are the two main types. We're going to focus on specifically ion channels, right? Ion channels. So remember that charged particles cannot go through the membrane easily. They need to go through a channel in order to do that. Now, we have three types of channels. I'm going to go through each one of them and then we'll use them for later on. The first one is a voltage gated ion channel. Then we have a ligand gated ion channel and a mechanosensitive ion channel. So let's get to know each one of these. First one is our voltage gated channel, right? Voltage meaning that they have some kind of a charge. So these voltage gated channels, they open and close when the membrane voltage changes, right? So our membranes can be either positive or negative depending on the presence of different types of ions. If we have more positive ions, then our membrane is more positive. If we have more negative ions, then our membrane is more negative. So depending on that, it'll open and close depending on the voltage. So some of the ions that we will see and we will learn about are sodium, potassium, calcium, and chlorine. So you can see most of those are positives and chlorine will be the negative, right? If you take a look at the picture, you'll see that it kind of has like this little cap thing where it can close the tunnel, for a little bit until, a change in the membrane potential again triggers it to open or to close. The next one that we have is the mechanosensitive ion channel. So same thing, mechano means movement. So these membrane channels are triggered by the movement of the membrane. So for example, this one gives us the mechanical force of pressure, right? So there again, the membrane is fluid, so it can move. So if there is pressure that is applied to the membrane, right, it opens up that channel, lets in whatever molecules are allowed to go through, and then once that pressure is released, then those ligands can't go through anymore, right? You want to think of it as kind of like a balloon, right? When you squeeze a balloon, usually the other side of the balloon inflates because of the pressure. So, in the same way, if you squeeze or input pressure in the membrane, the channel will open, and then when you let go, right, the channel will close back up, just like a balloon. The last one is the ligand gated channel, and this one is driven by an electrochemical gradient. So again, we're dealing with ions here. So the difference between this and the other types of channels is that a ligand does need to bind to the protein itself. So in this particular example, we see a neurotransmitter is stuck onto the channel itself, causing sodium to come in. And then when the neurotransmitter leaves, then the channel closes. Right? We will see this example again later on since we are focusing today on neural communications. But that's what a ligand gated channel is. So before we see these two things in play, I will again give you guys a review on the parts of a neuron. So this is a neuron, which is a brain cell, right? So we have our dendrites that kind of stick out of our neuron. We have the nucleus. We have the axon, which is where the signal travels through, and then the axon terminal, which is at the end of a neuron. So at the end of the axon terminal, there should be another dendrite there that will pass the signal on to the next neuron. So action potential, this is one of our learning goals, right? So what is an action potential? It is the signal that is moved through the cell, right? So again, it'll start at the dendrites, it'll flow down the axon all the way to the axon terminals, and then to the next neuron. And these action potentials happen really quickly. So the fact that I am thinking, the fact that I'm feeling and seeing and hearing myself all at the same time means that my action potentials are firing rapidly, right? And that means that my brain is working and it's functioning well. In each action potential, we have three main events. We have depolarization, which makes the membrane more negative. We have repolarization, which brings us back to the resting potential, meaning that there will be less negativity and more positivity. And then the last one is hyperpolarization, where we do go back to the resting potential. However, we go past the resting potential. And I'm going to tell you guys why that is important a little later. So here's a graph. I'm going to move myself again so we can see the entire diagram. So here's a graph of the steps that I just told you, right? And then with this graph, we see which membranes and which ions are participating and are causing that change in charge. So at resting potential, we're going to be right here in this beginning part. At resting potential, there is more of a negative charge. So our resting potential is, based on this graph, about 65, negative 65 millivolts. And then as we go and start our action potential, you can see that the charge on the membrane is becoming more positive, right? That means if you take a look at the key here, it's looking like there are more sodium ions coming in, right? And same thing, our potassium coming out. So we are experiencing that increase to the positive charge. When we hit the action potential, which is going to be this kind of incline right here, right? You can see again there are more of these sodium channels opening up, allowing that positive charge to manifest. And then at the end, it's going to drop down dramatically. That's the repolarization part, right? And you can see that now the only thing that moves outwards is the potassium ions, right? So one thing to notice about the membranes, see how that the red molecules are only going through the red molecules and the blue molecules are only going through the blue proteins, right? They can't enter the other way because the channel is not going to allow that. The only thing that is not shown here is the hyperpolarization, which don't fret because I have another diagram for you that will show that. So here's another I'll keep myself right there. But here's another look at the steps that I was telling you with a little bit more focus on the membrane charge of each of these intracellular, extracellular space. So number one, again, is resting potential. So if you can take a look at this side right here, we have positive on the outside, negative on the inside. Right? And then when depolarization happens, the voltage gated sodium channels will open, right? That's happening right here. It'll open and then it'll let the sodium ions through, which has a positive charge, turning the inside of the cell positive and the outside of the cell negative. Right, and then we hit the top and then go back down for repolarization. Notice the voltage gated potassium channel is now open, and again, this still works with the concept of diffusion, right? High concentration to low concentration. And since the opening of the sodium gated channel caused the influx of sodium ions inward, we have now a high concentration inside the cell. So now the in order to reach equilibrium again, the potassium gated channel will open and let those positive ions out. And then here's the repolarization, right? So see how we go way, way, way more negative than the threshold. This actually allows our neurons to stop the signal from going backwards. We want the signal to be moving forward to the next neuron. And this hyperpolarization is the one that allows us to do that. Okay, so now that we know how the signal transfers and the channels that are involved within each neuron, we're going to see what channels are involved when the signal leaves the presynaptic neuron and moves on to the postsynaptic neuron. So just to give you guys a little bit of an idea of where we are where those green circles are. We are leaving our main neuron where, let's say, the signal started, and then we are moving on to the next neuron who's going to receive the signal. So don't worry too much about all of those words on there. I'm going to explain them as simply as possible. I'm just trying to show you guys where we're at. Okay, so here's a better looking picture, a little bit more high def, but we have two main channels involved in this transition from the presynaptic neuron to the postsynaptic neuron. So as I've explained earlier, there was action potential, membrane voltage change all throughout the axon here, and now it's gonna go to the axon terminal, right? So when it reaches the axon terminal, right, the electrical signal will cause the voltage gated calcium channel, since it is voltage gated, it will allow that to open and allow an influx of calcium ions inward, right? This is signified by these purple protein and purple little circles. Purple circles are your calcium ions. So when that happens, right, the influx of calcium ions will actually encourage the synaptic vesicles to move towards the end of the membrane to release the neurotransmitters. So that's these circles right here. The orange circles inside are the neurotransmitters that will interact with the postsynaptic neuron, right? So once these synaptic vesicles fuse with the membrane, they will release their neurotransmitters, and then this is where the ligand gated channel comes into play. So as you can see down here, right, these orange little balls are interacting with the receptors on the other side, right? And since it is a ligand gated channel, when that interacts, it opens, right, allows the sodium ions to go inside, which then ultimately gives us a nerve response, right? A nerve response. Um, Something to pay attention to, again, neurotransmitters, right, can give us different effects depending on what kind of neurotransmitter it is. It can give us an excitatory response, or it can give us an inhibitory response, just depending on what type of neurotransmitter is being released at that time. So now, let's get to the last part of our learning goals, which is to figure out how do the membrane, right, the neurons, the proteins, how do they all work together in terms of signal transduction? So again, just a quick overview of a signal transduction. Remember that there's three main parts. We have reception, transduction, and amplification. That's the same for any kind of signal transduction, regardless of what kind of protein receptors are involved. So let me give you guys a specific example when it comes to the neuron itself. So in this particular diagram, again, it's more of a simplistic diagram, but you'll have some kind of a signal molecule interacting with the receptor protein, right? Most of the receptor proteins will be on the membrane, so whether that's G protein or RTKs, right? They will be on the membrane itself. They're going to transduce that signal through a phosphorylation cascade, and then at the very end they will amplify that signal with the use of a variety of things, including second messengers if that is involved. So here is the main example. So the example I chose to talk with you guys about is the nerve growth factor, or NGF. It's the one responsible for the differentiation, survival, and synaptic connectivity of sympathetic and sensory neurons. So what it does, right? What it does, that is, if we go back a slide, that is going to be our signal molecule. So it's going to bind to a tyrosine kinase receptor, right? And in our case, that'll be this purple protein right here. So when NGF and the TRKA bind together and they interact, they're going to move on to the second step, which is again the transduction, and this is caused by the phosphorylation cascade. And then at the very end, they will produce IP3 and DAG, which is types of second messengers, right? From there, those second messengers can do a lot of different things. This particular example that I chose stimulates the release of calcium from the endoplasmic reticulum. So think about what we just talked about. Remember that calcium is the one that helps stimulate the fusion of the secretory vesicles onto the edge of the axon terminal. So if you increase those levels, then that means that the secretion will also be quite increased. So again, depending on the neurotransmitter, here's the picture again, right? So NGF will be somewhere around this area as well, because this is where the influx comes in, right? So NGF binds to TRKA, right? Releases all that calcium. Calcium will probably go through the same voltage gated channels that we see here, and then the synaptic vesicles will then release the neurotransmitters. So let's say, for example, that your neurotransmitter causes you to have an inhibitory response. So let's say, something like a depressant like alcohol or drugs, right? If there is an increase of that NGF interaction with that receptor, there's going to be even more inhibition than your normal response would be because of that increase in calcium ions, right? So all of these work hand in hand together to elicit a response, and depending on what molecules are involved, then we get a stronger response if we have, for example, NGF involved. So that is the end of our presentation. I would highly suggest that you guys go back and take a look at the learning goals. Make sure that you feel comfortable doing those tasks so that we know and you know specifically which areas you need to improve on. And if there are some areas where you still feel kind of iffy, I would say to rewatch the video and solidify your knowledge of the topic even more. But thank you guys for watching the video, and I'll see you all next time.